We're going to start the chapter on enzymes, which is the video 2, in which we're going to discuss investigate. And this is syllabus code 5090, and this is the biology for CIE, and this is the O level. And this is the second video on the chapter on enzymes. Now, as you look at the syllabus is enzymes, enzyme action, temperature, and pH. And the D part is which we need to talk about: investigate, describe the effects of temperature and pH on enzyme activity. So whenever there is an investigate, we need to discuss the practical procedures. How are we going to investigate? How are we going to do this in the lab? So we're going to be talking about how we're going to do this using laboratory apparatus. Now, whenever we investigate, we have to use practical procedures and we have to use laboratory apparatus. Now, you must be knowing the names of all these different laboratory apparatuses, because that is only when you are comfortable and you can then be able to do the paper 6 questions as well. So, we're going to now look at these different laboratory equipment and how we name them and how we use them. Now, here is some of the laboratory equipment that we usually use, and the names must be familiar to you, and the spellings must be also correct. The first one is beaker. So, a beaker is of different sizes. Then we have test tubes. Then we have the Bunsen burner. Then we have the mortar and pestle, watch glass, goggles. Then we have the burette, the pipette, thermometer, the graduated cylinder, the water bottle, spatula, funnel, file, forceps, lab burner, test tube holder, micropipette, tongs, wire gauze. So basically, main ones which we use are the beaker and the test tubes. So you must know and what do they look like and how do we use them. And we, of course, we have a test tube rack as well in which we place the test tubes. So you must be familiar with these names, you must be familiar with the spellings, and you must be able to mention them in the practical procedures whenever we do those practical procedures. Then we need to talk about the iodine test, which we're going to be doing in this. We use it to test for starch. How do we make the iodine? We dissolve the iodine in potassium iodide. So it is a potassium iodide solution. Then we take whatever sample and we add the iodine solution and directly to the food sample, and you get a dark blue black color indicating the presence of starch. So this is the color of iodine, which is a brownish color, and then it changes to a blue black color, which is here, which you can see here. Now the intensity of the blue black color, either is a dark blue or a mid blue or a light blue, it gives you the quantity of starch present. So it does also tell you briefly, but a very rough estimate of how much is the starch present in that sample. Now let's investigate the effect of temperature on the rate of enzyme action. Now what we have to understand is that there are two things here: temperature and the rate of enzyme action. Now one of them is classified as the independent variable. Now things which we're going to change, we are going to change the temperature. We're going to decide, okay, what temperatures are we going to have? 30 degrees, 35 degrees Celsius, 40 degrees Celsius. So if the thing that we are going to change, that is classified as an independent variable, and we usually take 5 of those. So we will take 5 temperatures: 30, 35 degrees Celsius, 40, 45, and 50 degrees Celsius. So we will take 5 different temperatures to able to study the effect of temperature on the rate of enzyme action. So the thing which you change is the independent variable. So independent variable, then the rate of enzyme action, this will be the dependent variable. And this is we're going to measure by somehow. It must be in time or maybe in number of bubbles or something else, which is going to assess how fast the enzyme action is taking place. Now we will take 5 test tubes and we will keep them in 5 different temperatures. And how will we keep them at 5 different temperatures is we'll take a water bath and we'll add water to it and we'll add a Bunsen burner to it and we will place a thermometer in it so that we can then record the temperature and see that the temperature is maintained at 30 degrees Celsius. So a beaker, this is indirect heating. This is a beaker with water in it. And at times we give you hot water to put in it so that it doesn't take a lot of time to heat that. And then this is a thermometer in it to maintain the temperature so that you can see how much is the temperature. Is it going up? Is it going down? So we will adjust the flame of the Bunsen burner or we'll close down the Bunsen burner to maintain the temperature at 30 degrees Celsius. Now, what are we going to use? Now, I've given you an example. We are giving you a 1% starch solution and we have given you a 5% amylase solution. Of course, this can be different. It can be a 2% starch, can be a 10% starch. That's immaterial. But whatever it is, it has to be the standard which we are going to be giving you. So when we're giving you a starch solution, we have to first decide the volume of it. The volume must remain the same in this situation. Usually the volume, the total volume of this should be the same. So we're going to take 5 ml, 5 ml 1% starch solution. Now this will be in all, it will be 5 ml. All of them will contain 5 ml. So 5 ml starch in each of the 5 test tubes. And then let's take 5% amylase. Now the 5% amylase, we're going to say take 10 ml. So 10 ml of the amylase. Now all of this will have 10 ml of the amylase. So 10 ml amylase here, 10 ml amylase here, and 10 ml amylase here. Please remember, but we are not going to mix them, all of them at one time. We'll do them one at a time. So let's do the first one, which is the 30 degrees Celsius one. But just to standardize it, I want you to understand it that the volumes must remain the same. So 5 ml starch and 10 ml amylase. You could have said 10 ml both. That's fine as well. I'm not saying that that's the only one you've got to take. You could take 10 ml amylase as well. So I'm fine with it if as long as you specify a volume and the volume must remain the same. Now, why are you doing this? The reason why you're doing this is you're ensuring that the enzyme is, say there are 1,000 active sites or there are 10,000 active sites. So all of them have 10,000 active sites and then all of them have the same number of the starch molecule, which is say 100,000. So the number of enzymes is the same and the number of starch molecules, which is the substrate, is the same. So you have standardized the procedure and you have ensured that what you are going to change, you're only changing the temperature. But the number of starch molecules and the number of enzyme molecules, which means the number of active sites, are the same in all these 5 test tubes. Now, how are we going to study the rate of enzyme action? You see, in the rate of enzyme action, what we have to study is how long does it take for the entire starch to be converted to maltose? All the starch should be converted to maltose, so there should be no starch left. Now this time we have to find out. Now this time is the one that we've got to figure out. So we need a stopwatch for this. Now when the stopwatch is when we have mixed the two things, we mix the starch and the amylase. As soon as we mix it, we take a drop from here. Now what we need to do is before that is that we take a white tile and we place drops of iodine solution in that. So there's iodine solution here, iodine here, iodine here at a short distance from each other so that they don't mix with each other. We place these drops of iodine. And now as soon as we mix this, we take a drop from here and we mix it with the iodine on the tile. This was at the first one was at 0 at as soon as we mixed it, so 0 seconds at that time, and we see that the color is black. This means starch is present. Now after another 30 seconds, first we took it at 0 as soon as we mixed it, then we take it after 30 seconds and we do the same thing. We take a drop and we mix it with the next drop, and the color is black. And we do it after another 30 seconds, black. So after another 30 seconds, we take another drop from here, and like this we keep on doing this, and it's black, then it's black, still black, brown. So black, 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 and finally we find out that it remains brown. Now if it remains brown, it means that the starch has been converted to maltose and no more any starch is left. So this was at 0, this was at 30 seconds, then another 30 seconds, so 30, 30, 1 minute, 1 minute 30 seconds, 2 minutes, 2 minute 30 seconds, 3 minutes, and 3 minutes 30 seconds. So it took 3 minutes and 30 seconds to convert the entire starch to maltose. Now 3 minutes means multiply this by 60. Now this would be 180 seconds plus the 30 seconds, and this will be 210 seconds. So it took 210 seconds for the entire starch to be hydrolyzed or to be digested. Now we will repeat this experiment at different temperatures. So then we will repeat it at 35 degrees Celsius, then at 40 degrees Celsius. Now at 35 degrees Celsius, what is going to happen? This should be less time. Why less time? Because as you have increased the temperature from 30 to 35 and 35 to 40, the kinetic energy increases. So the rate of enzyme reactions. So it will take less time. As we increase the temperature, it should take less time. I mean 30 to 35. But of course, then we don't know the optimum of that enzyme. We don't know if it's a human enzyme. We don't know if it's some other enzyme. But the general rule would be if the temperatures are below the optimum, then of course, as you increase the temperature, it would take less time for the starch to be digested. Now the same tile story, we will have to repeat it at 35, then we'll repeat it at 40 degrees Celsius, then 45 degrees Celsius, and 50 degrees Celsius. Now we will get the data for 5 different temperatures, and then we can plot a graph of this data. But we can also do this is repeat 5 times at 30, then repeat 5 times at 35, then repeat 5 times at 40, then 5 at, so we will be doing 1, 2, 3, 4, 5 into 5, 25 times this practical to just to get a reliable result. Why reliable result? Because when you do it 5 times, then you know the trend. So you know, okay, it's taking so many seconds, and that gives you the trend, and then you can calculate the mean, and you will get very, very reliable results. Now a student plotted a graph, and as you can see, we put the independent variable on the x-axis and the rate of enzyme is on the y-axis. And this could be in maybe arbitrary units, which is a general term we use for random units, and we talked about this in a previous video. So arbitrary units. Arbitrary means there's no proper unit to it, but it's just some units that we have attributed. Now a student might get a graph like this, and he or she assumes that what is happening is that as the temperature is increasing, the rate of enzyme action is increasing as well. and he gets a graph like this. But what has he done wrong? He has taken temperatures which are below the optimum, and he assumes and he thinks that okay, well, as we increase the temperature, the rate of enzyme increases. But then he only took these temperatures, this one, and this one, and this one. But when he takes more temperatures, he realizes that the graph changes. So initially, if he got a graph like this, and he assumes that it is a linear correlation, that as the temperature increases, the rate also increases, but then he has made a mistake. He's taken temperatures which are much below the optimum. He needs to take more temperatures, and then he sees he gets a graph like this. So what you have to understand is that it depends on which temperatures did the student take. Did he take temperatures which were below the optimum? Of course, then he gets a graph which is just a straight line. But he needs to take a wider range of temperatures so as to assess what could be the possible optimum of that enzyme if he is trying to find out what is the optimum of that enzyme. So this is how we would plot a graph of temperature against rate, and we would get a graph which is like a graph like the one which I've just drawn. An important improvement could be made on this practical is that you initially, we just said, okay, take starch and amylase and add it in the test tube and then keep it in the water bath. Well, that's incorrect, because if you have added the starch and you have added the amylase, well, the reaction has started. And if this reaction is in a room which is hot or cold, we don't know. If you're doing it in June, this room is going to be very hot. But if you're doing it in December, it's going to be a rather cold lab. And then you say, okay, let's then again put it in a water bath at 30 degrees Celsius. Well, that is wrong, because what has happened? The reaction has started. If the room was hotter, then the reaction started at a higher temperature. If the room was cold, well, the reaction started. And then you say put it in a water bath. Even if you take a glass of water from the tap and you put it in the fridge, it takes about 5 minutes for it to cool down to 4 degrees Celsius. So what you need to do is that this is an improvement on this practical, is that you need to keep them separately first. So the amylase is separate and the starch is separate. And you keep this for 5 minutes so that both the amylase and the starch are at the same temperature. And you keep this in a water bath at 30 degrees Celsius. And then after 5 minutes, you mix the two. And then, of course, the reaction would be at 30 degrees Celsius, because you kept the starch and you kept the amylase separately. So both achieved the temperature, and then you mix them so that the reaction would actually start at 30 degrees Celsius. So this is an improvement which you must know on this experiment. Next, investigate is the effect of pH on enzyme action. Now for that, I am using the same experiment, but I'm modifying this now slightly. So what I have ensured is that the volume of starch is the same in each. So the volume of starch is 5 ml in each. You can have it 10 ml if you want to, but I mean, it's up to you. You have to standardize it. So same. Then you added the same volume of amylase to each. So amylase is the same 5 ml, 5 ml, 5 ml. Please remember, I'm just drawing it to make you sure that we're going to do it in 5 different test tubes, but you have to start with one at a time. And now, in order to change the pH, you know what is pH? pH is the power of hydrogen. So you need to add something to it. But the important thing which you've got to remember is that it must be the same volume. So whatever you're going to add has to be the same volume. So it has to be 5 ml. So the total volume in each test tube is 15 ml. Now you can't say add 2 ml to one and then 3 ml to the other and then 5 ml to the other and 6 ml to the other. The total volume is going to change. So whatever we are going to add, we're going to add 5 ml to each. This is what we need to understand. Now if we look at the pH ranges, so we can have 7 here, we can have 9 here, we can have 11 here, and we can have 3 here and 5 here. So, but in this now, this 5 ml what is this going to be? In this 5 ml is going to be distilled water. So the pH will remain neutral or 7. Now in this one we'd have to put some alkali. So let's put some KOH. And here also KOH. But then if we're going to give 5 ml, we've got to realize is this will have to be 0.5% KOH and this will have to be 1% KOH. Now here we're going to add HCl, dilute HCl. Here also we're going to have dilute HCl. Now this one, to keep the pH at 3, will probably be a 1% dilute HCl and this will be a 0.5% because 3 is highly acidic. So it have to have more HCl in it. Now once we've done this exercise, in order, now this is for you all to understand very clearly, is that we kept the starch the same. Starch volume was the same. Amylase volume was the same. Now what we change is, in order to change the pH, we either added HCl or we added an alkali, which can be sodium hydroxide or potassium hydroxide. And in this we've added distilled water. But the important thing is that we added the same volume. So the total volume remains 15 ml. Now what we will do is we'll do the same story, that we take a white tile and we add drops of iodine solution to it at spaced out intervals so that we don't want it to mix. And then what we will do is we will take a drop from here and then mix it with the iodine, and the color remains black. It means that starch is still present, and we keep on doing this after every 30 seconds. So after some time we will find out that it remains black. Every 30 seconds, black, 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 and finally it is brown. So we add up the 30 seconds. So this was at 0. So 30 seconds, 31 minutes, 130, 2 minutes, 230, 3 minutes, 3.5 minutes. So it took 3 minutes and 30 seconds for the entire starch to be hydrolyzed. So we would do this is for the different pH. Now if you look at these five test tubes that we had, the important thing is that you would then keep all of these at the same temperature. Now you're not going to take the temperature. So the temperature will be, in this case, was maintained at 30 degrees Celsius. So the temperature for all of these would be kept in the same water bath, or we would do it at the same. I won't say you do them all at the same time. You would have to space them out, and you'd have to do this one first, and then this one, and then this one, and then this one, and this one. So you would then use the data. You would then use the data to plot a graph. And on the graph, you would have the pH, which is the independent variable on the x-axis. And the rate of enzyme action would be on the y-axis. And you would get a graph something like this. So pH on the x-axis and rate of enzyme action on the y-axis, and you would get a graph, and you'd plot your results for the time it took. So because the rate of enzyme is the time it took for the entire starch to be hydrolyzed. So this would be, the time would be in seconds. And you would plot a graph of this, how quickly it took. So the lesser time it took, that would be the optimum pH of that enzyme. Now the last thing that you need to do with every chapter is, once you've completed the chapter and you've made your notes and you've gone through the book, you will start doing the past paper questions. But before you do the past paper questions, you need to again look at the syllabus points which you need to cover. And you need to be very careful about going through each and every point. Like the first point was define catalyst. Now this is a chemical catalyst. They expect you to know what is a chemical catalyst, that speed up chemical reaction, not changed by the reaction. Then it says define enzymes as proteins that functions as biological catalysts. So you must know what is a normal catalyst in chemistry which you study, and one which is a biological catalyst, and those have to be proteins. And then explain enzymes in the term of lock and key hypothesis, And then this is the investigate and describe the effect of temperature and pH on the enzyme activity. So it's a very good habit to study is to go across, and after you finish the chapter is to then again reassess the syllabus and see if you have understood each and every point. And then of course you need to mark the point, say if this is not clear to you. So then you need to go and see your teacher and ask her or him that, I'm not clear on this point. Can you please explain this point to me? So I'm sure all of you will do a good job at this, and best of luck, and thank you.